So the first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases together. What is also used is topoisomerase, and it relieves the overwinding strain ahead of replication forks by breaking, swiveling, and joining DNA strands. The separation of the two single strands of DNA creates a Y shape called a replication fork, and the two separated strands will act as templates for making new strands of DNA. Connected on the two single strands are molecules of single-strand binding proteins, which stabilize the unwound template strands. One of the strands is oriented in the 3 to 5 direction, and this is called the leading strand, and the other one is in the 5 to 3 direction, and this is called the lagging strand. As a result of their different orientations, the two strands are going to be replicated differently. So the leading strand, a short piece of RNA called a primer, produced by an enzyme called primase, is going to come along and bind to the end of the leading strand and acts as a starting point for DNA synthesis. The DNA polymerase binds to the leading strand, DNA polymerase, and then walks along it, adding new complementary nucleotide bases to the strand in the 5 to 3 direction. In the 5 to 3 direction, yes. And this is going to be continuous. And so on the lagging strand, numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Okazaki fragments are then added to the lagging strand, which are also in the 5 to 3 direction. And these are discontinuous, as the Okazaki fragments will need to be joined up later. Once all the bases are matched up, an enzyme called exonuclease strips, the strips away the primers, and the gaps of the primers are filled by yet more complementary nucleotides. And finally, an enzyme called DNA ligase seals up the sequence of the DNA into 2 continuous double strands. The result of DNA replication is 2 DNA molecules consisting of 1 new and 1 old chain of nucleotides. And this is why DNA replication is described as semi-conservative, with half of the chain part of the original DNA molecule and half brand new.